Meiosis was discovered and described for the first time in sea urchin eggs in 1876 by the German biologist Oskar Hertfig. Sound familiar? Or actually, it was discovered for the first time in horse intestinal threadworm in 1883 by Belgian embryologist Edward van Beneden. In either case, we will get to that briefly and figure out which one is correct. In experimentation, it is incredibly important to make careful observations. In the process of science, experimentation always begins with observations of some sort. This leads to a hypothesis of some sort, but then gets evaluated through further observation. In many of the natural sciences, such as the physical sciences, they rely on quantitative forms of data. However, the life sciences are unique in how it can replace the need for data in investigations with observations. Okay, I guess this would count as qualitative data. It is pretty spectacular, though, how biologists of all kinds can use observations to evaluate a hypothesis. Take animal behavior, for example. Jane Goodall, English primatologist, ethologist, anthropologist, and UN Messenger of Peace, has been studying chimpanzee social and family behavior for the last 55 plus years. Her meticulous journals and data sheets, though recording time, is heavily laden with detailed descriptions of the behaviors, actions, and interactions between individual chimpanzees in the troop. Because of her work, she gained community cooperation from villagers around Gombe Stream National Park, Tanzania, to create a buffer zone to protect the chimpanzee sanctuary while still being able to use the land for their livelihoods. She has gotten the National Institutes of Health to reevaluate chimps in scientific research, which led to the retirement of 310 chimps in 2013. This is the tip of her legacy. Can you imagine how different the world would be if we were all half as careful with making and recording observations? Which brings us back to our original debate. Who discovered meiosis, Hertfig or van Beneden? Oskar Hertfig was studying sea urchins on the Mediterranean. Sea urchins are an excellent model organism to study within reproduction for several reasons. Sea urchins produce eggs for reproduction in very high quantities. These eggs are released into the water, and post-fertilization events can be observed with ease. These eggs are quite large, on average 100 micrometers in diameter, and the sea urchin eggs have a clear cytoplasm which allows to clearly see intracellular activities. Due to his impeccable observations, he observed that fertilization involves two germ lines, and that the nuclei of each united, where the female gametes persisted. So Hertfig became the first to discover and describe the idea that meiosis existed, but he didn't have an explanation or observation for the process. Edward van Beneden read the research published by Hertfig and developed his own methodology to understand what was going on. Rather than using urchins, he opted for Ascaris megalocephala, a roundworm that infects the intestines of horses. The greatest advantage to this is the female reproductive system is designed for reproducing a lot. Basically, from end to end, you can find eggs at some point of development. That made it easy to observe under a microscope and carefully draw out each stage of the process. And so meiosis was discovered and described at the chromosomal level. Fun fact, meiosis is derived from the Greek word meaning lessening, as in having the number of chromosomes. Get it?